So, who have sent the slide, they can look at the slide and explain. Understood or not? Those who have not, they will write the heading and then explain. But it is mandatory, not only compulsory, also mandatory to prepare slide and send. Understood or not? Don't bite nails. So, without delay, let me welcome on the stage Sarnia Bumrel to present in her presentation. Okay, I can see the.
Role of DNA test in different researches. DNA is extracted by different agencies for different purposes. Some of the purposes are here. Forensic teams use DNA to identify suspects, eliminate them from consideration. It can it can establish a person's innocence or guilt and determine if they were present at crime scene. Next purpose is paternity tests. DNA extraction is valuable in determining the paternity of a child, whether confirming or refuting paternity claims. DNA from both the potential father and the mother provides evidence. And similarly, there is next purpose, ancestry tracking. Beyond identifying immediate ancestors, DNA extraction helps individuals to understand their ancestors' origins. Modern DNA kits reveal countries of origins, living relatives, and potential predisposition to medical or food conditions. Fourth, fourth purpose is medical test. For certain medical conditions, DNA extraction is essential for official diagnosis, especially when the condition is genetic. DNA extraction also identifies carriers of genetic diseases. Thank you.
Okay, thank you. So, actually, you will have what about DNA test, right? So, DNA test is applicable in different activities. So, number one is here forensics. So, you are very much famous about those CID, right? Yeah. So, when there is to be some problem, they have to send those items in forensic lab, right? In forensic lab, what is the name of doctor? Doctor Salungi. Salungi doctor, very powerful doctor, right? Very, yeah, identify everything. That means he had technique to match or mismatch the DNA, right? There was crime, and whether Mr. Nupani was present or not, so for that they will collect some cells, they extract the chromosomes, and then they tally. If it matches, he was present. If not so, not present. That is forensic one. Next one is paternity test. Sometimes there is compulsion, bro, whether the child is of that parent or not. In that case also, so DNA test is done, right? If the DNA matches, then definitely he or she is the parent of that person, that baby. Otherwise, not. Next one is ancestry tracking. Ancestry tracking means actually our ancestors from where they have come, right? Suppose if you talk about me, my ancestors were not of this place. They were from Bhojpur Gogani. So another person, like there are two persons, they say, they clash. Oh, our ancestors were there and we came from same place. So for that, again, they also have to do DNA test. If it is matches, then yes, definitely they were from same ancestor. Otherwise, not. And what is next one here? Medical test. So for diagnosis or finding some results, they just carry out this DNA test. Okay. So Sarnia's presentation was very good. Give her a big round of applause. Now it's the turn of which one? Which turn?
Nice, sir.
Please come. So I have just made this one. Do you have?
Good afternoon, everyone. It's Mr. Sanwal. Today I am going to explain about role of DNA test in various researches. First role is genetic engineering. Genetic engineering uh, helps in genetically engineering plants and animals. In plants, generally, it helps to identify, isolate, and uh, extract desired genes of a plant. In animals, it plays a very crucial role from cloning to transferring gene of one animal to another. Second role of DNA test is vaccine. DNA also contributes in production of vaccines, as it is not fully approved for human use. It is generally used in animal vaccines and in some general development of human vaccines. Third role of DNA test in various researches are hormones. DNA extraction of DNA helps in developing hormones. Some hormones that use DNA extraction are human growth hormone and insulin. Human growth hormone is a type of hormone which is responsible for the overall growth of body. Insulin, generally diabetic patients require insulin, generally in type 1 diabetes, as DNA extraction also help in development production of insulin. It is very beneficial. Thank you, everybody.
Thank you very much. So, though he did not make slide, but he explained these three things. What are they? One is in genetic engineering. Next one, production of vaccines and making different hormones, growth, human growth hormones, and production of insulin. See, we have got one part in our body. It is called pancreas. What do you call it? Pancreas. What is the main function of pancreas? To produce insulin, right? So what does insulin do? It decreases the sugar level. It decreases the sugar level. We eat different type of nutrients, carbohydrates, protein, vitamins. You keep that ballpen down. Minerals, fat, water, raw phase, etc. Mainly carbohydrate provides us energy. So we eat more and more food which provides us carbohydrate. But if there is excess such food in our body, then it cannot be utilized. Yes or no? Then it moves along with blood, and there is problem. We call it diabetes. So to reduce that sugar level, insulin should be produced. But if the pancreas does not function well, if the production of pancreas is very less, at that time, the problem of diabetes increases. So the diabetic patients are provided with injection in the that insulin injection in the form of liquid. So that is prepared by the DNA. Okay, thank you very much. Now who's turn? Who's turn now? Simresh turn. Okay, Simresh.
Not selective breeding. The term artificial selection was coined by Charles Darwin, and selective breeding is one of the most earlier form of biotechnology. For example, we can see a dog. Many scientists believe that the dog were evolved from wild wolves, and humans have been researching about dogs about 14,000 years ago, and it is one of the earliest form of biotechnology. And selective breeding can be used for producing tastier fruits, vegetables, and for large animals, which can be used as meats. Selective breeding is one of the most important in biotechnology. Thank you.
Thank you very much. So selective breeding means good characters are chosen, bad characters are left there, isn't it? So the biologists, the researchers, they just keep on searching the main factors which give higher range, and those characteristics are taken, chosen, and enhance the quality of food, fruits, vegetables, etc. Right? Okay. Thank you, Simres. I'm not saying. Now who's turn? Ritika's turn. Okay, Ritika is here. Yeah. Oh, Ritika's slide is there. Okay. Ritika's slide is there.
Namaskar everyone. Namaskar everyone. It's Ritika Bharat. Today I am here to give the presentation on genetic and genetic technology. Selective breeding. Scientists believe that domestic dogs evolved from the wild gray wolf through artificial intelligence. Human were able to create hundred of different dogs breeds. Likewise, as people domesticated and breed dogs over time, they favored specific traits like size or intelligence, such as hunting, breeding, or championship. As a result, many dog breeds have vast, vastly different appearance. Selective breeding has also been practiced in agriculture for thousands of years by isolating wild cabbage plant with specific characteristics. Farmers were able to create a varieties of vegetables for a single source, each with different flavors and texture. Similarly, it involves choosing parents with particular characteristics to breed together and produce offspring with more desirable characteristics. Thank you.
Thank you very much. So this what do you say? Slides presentation. It is the best technology. Understood or not? So nowadays, why are you biting that ball pen? Are you nursery kid? Asonish? Remember it? Keep your hand down. Relax. Provide support to hand and relax. Back one straight. Lazy bones are here. So, how efficiently she has done? So, see here. Earlier there was only wolf. What was there? Wolf. So, from wolf, different type of dogs are produced. See, in Europe, a kind of dogs are produced. In America, next type. China, next type. India, next type. See, environment also affected them, and genetic factors also affected. Now, it, if you go in that kennel club, we can get different breeds of dog. Yes or no? And in Nepal, we don't have here Tibetan Mastiff. You might have heard its name. Actually, that is for shepherding. So it is used for hunting also, right? So very powerful dog. And I heard its price also very high, not less than crores of rupees. And some dogs, American Bulldog, I found one of my friends, he was using American Bulldog. He has paid 90,000 rupees for a small puppy. See, LCCN is also there, German Shepherd you can say, and Japanese species there, species. Next, Husky is also there, right? Different brands are there. Once they were same, but due to this genetic engineering, their selective genes were taken and chosen. Okay, so not only in animals, this technology can be done in plants as well, right? See, number of people are increasing, but the land is not increasing. That's why what have the people thought? They have thought, why not to produce more advanced crops? Nowadays, they have got seed of particular plants which can yield more and more crops, more and more fruits. That is because of selective breeding. Okay, thank you very much, Ritika. Now, whose turn? Luniva. Luniva's presentation is there or not? Good. So they are smart. They are smart and
Namaskar and good afternoon to you all. As you all meet me, Luni Ramananda, to give presentation on the topic of method of selective breeding. There are two methods in selective breeding, interbreeding and outbreeding. Interbreeding is the method of selective breeding in which breeding is made in between sire male and dam female of related animals or related breeds of plants. Siamese cat and Labrador dogs are some example of pure breed. Outbreeding is a method of selective breeding in which breeding is done between unrelated animals. There are two types of outbreeding: crossbreeding and grading up. Crossbreeding is the mating of two different breeds. Grading up is the breeding of two different breeds. Thank you.
Okay, thank you. All right. So, you go there. Now listen. Methods of selecting breeding is here, right? So, selecting breeding means selecting best qualities. Understood or not? So, Albert Einstein is not present nowadays. Otherwise, from his cell also they could extract best characteristics, best genes, and keep in new organism. New organism means new human beings, and more advanced human beings could be produced, right? Okay, so there are two type of selective breeding. What are they? Inbreeding and outbreeding. In means inside, out means outside. So in inbreeding there is cross between male. Actually male is called sire. You say sir, no? Male is sire and female is dam. DAM, dam. No, here is given dome. It should be dam. Look at your book. Dam. Madam. They say, no. Hello, madam. Yeah, madam. Sire and dam of related animals. Same related animals it is done. And what are the examples? Siamese cats and Labrador dogs. In outbreeding are of how many types? Two types. What are they? Cross breeding and grading up. What is done in cross breeding? Mating of two different breeds, and here breeding of two different breeds. Here mating, here is breeding. Right? So now whose turn? Okay, Nilisa's turn. Nilisa, do you have any thing or not? No slide? Okay. Now you can explain. Oh, you can make everything up to date. Hey, students, be smart and make every day minimum 1 or 2 slides. So presentation will be easy. Understood or not?
Namaskar, it's me, Jestha from grade 10. I'm standing here to tell advantage of crossbreeding and disadvantage of crossbreeding. Advantages are it promotes a fast growth rate by accelerating. Cross breeding boosts biodiversity by increasing the varieties of organisms. Disadvantage of crossbreeding result in the loss of original genetic traits. There is possibility for crossbreeding to experience a lower fertility rate. Thank you.
Okay, thank you. Actually, everything has got advantage as well as disadvantage. So here are advantages of crossbreeding. Here are disadvantages of crossbreeding. What are the types of crossbreeding? Inbreeding and outbreeding. Remember. So, what are the two advantages? It promotes a faster growth rate by accelerating those activities. The organisms can grow very quickly. If they grow fast, they give the result very quickly, right? What is second one? Cross breeding boosts biodiversity by increasing the variety of organisms. So they produce new organism and there will be biodiversity, diversity in plants and animals. New species will be grown because of crossbreeding. But disadvantage is crossbreeding results in the loss of original genetic traits, locally available things. Plants and animals, they will disappear because in the name of bombing new and new, it's gone. Like our costume,
Yes.
our culture, it is also being weaker day by day, or we are focusing on imported culture. That is also not so good. In Western culture, crossbreeding also like that. Because of continuous crossbreeding, our local breeds will disappear. Their genes will disappear. Next one, there is possibility for crossbreeds to experience a lower fertility rate. Yeah, sometimes those crossbreeds, new organisms found, they may not produce more offsprings. We have read about liger and tigon. Liger and tigon, they were produced, but they could not produce new organism, mere offsprings. Fertility rate is 0. Okay, so that is all for today. What did we discuss? Remember,
Role of DNA.
role of DNA in different research activities. Number second, selective breeding, types of selective breeding, or methods of selective breeding. Next one, advantages as well as disadvantages. So thank you very much for the presentators, and thank you very much for the students who listen very carefully. Once again, give a glance upon it, what you have studied. Give a big round of applause for them also. Read. Now read, then afterwards I will give you some classwork also. If that not possible to complete, that will be your homework. Understood or not? Note making, it is regular. Now you are in grade 10, some sort of pressure should be there for study. Understood or not? Okay, thank you.